Parkinson's disease, you have a buildup of clusters of protein that's normally functioning well, but it clusters up and it goes bad. It forms these clusters inside brain cells. We need our brain cells to communicate to each other. We need our brain cells to function. But when you start to get this buildup over time of these clusters of bad protein, that leads to cells starting to die in Parkinson's, which leads to the symptoms that we see in Parkinson's disease. So what the Stanley Fahn Jr. Faculty Award has allowed me to do is to be able to understand, first of all, not what is the structure of these protein clusters, where are they starting to form, with what compartments are they associating with, because all of this leads to understanding how are we going to design better therapies, right? We need to know what's going on wrong and where this is happening. And we have tools to be able to tag and visualize where that's happening in the cell for really high resolution imaging in a very natural state using this technique called cryo-electron microscopy. And cryo-electron microscopy was awarded the Nobel Prize in 2017 and really allows us to look in a very natural state at extremely high resolution inside these cells. Now for the Impact Award, that bridges the cells that have these bad protein clusters to the atomic level, so really, really high resolution. So now we're going deep inside the clusters. We have a tool, a very small molecule from our collaborators at Harvard who screened many, many different compounds to figure out which ones bind very selectively to these bad protein clumps. And we're trying to figure out exactly at the atom level resolution how this binds to the bad protein clumps. We're trying to determine what is the interface. And based on that, we are designing new fluorescent or markers, basically, to then be able to not only track the progression from early stage to late stage in these cells, but also in living human patients. So a PET tracer is basically a marker. Think of it as a fluorescent marker that can be injected to living patients and can go very specifically to the type of protein cluster that's wrong, that people want to be able to track. Clinicians need to track for the progression of the disease, right? So in Alzheimer's, you would track the protein called tau, and there are many established PET tracers for that. We don't have that for Parkinson's disease. We do not have a PET tracer that reliably marks these clusters of the protein alpha-synuclein when it goes bad. So we can't tell when these clusters are starting to grow, when they're starting to spread in the brain, etc. It's largely relying on clinical diagnosis, symptoms. So having a PET tracer for synuclein in Parkinson's, it would allow clinicians to detect pathology earlier, monitor how it spreads over time, and evaluate whether experimental therapeutics are actually reducing the clustering and this accumulation over time. Importantly, it would also allow clinicians to be able to distinguish Parkinson's disease from other neurodegenerative conditions that have overlapping symptoms. The support from the Parkinson's Foundation has been absolutely catalytic, not only in terms of the funding, which has been absolutely instrumental for me to make the leap from looking at human Parkinson's brains to cell models to understand the early stages of aggregation and looking at how small molecules interact with these problematic protein clumps, but also the community. I feel every time I attend one of the Parkinson's meetings or interact with people, this sparks some kind of collaboration, or what I have to offer, what insights that I have help somebody else in the field, and we're all aligned on synuclein and Parkinson's disease. So it's a very special community, and I feel we're able to make real progress because of the funding, but also this interwoven community of people who really support and want to further Parkinson's research to ultimately cure Parkinson's disease. There's real momentum in Parkinson's disease research right now. We understand more about the problematic protein alpha-synuclein now than we did a decade ago. Structural biology has revealed atomic level details of this problematic synuclein protein. The cell models are becoming increasingly sophisticated, and there are newer imaging agents and disease-modifying therapies on the horizon. My work in particular brings hope because we're able to now look at these problematic protein assemblies in a totally natural state at extremely high resolution to figure out exactly where they're going wrong. Your investment is not abstract. It is helping to support experiments right now in real time that helps towards diagnosing and treating Parkinson's disease. You are accelerating and empowering young scientists like myself towards the shared common cause of curing Parkinson's disease.